So the lab is developing new techniques for delivering DNA vaccines. And these are very new and powerful types of vaccines. They're similar to the mRNA vaccines that were developed for COVID during the pandemic, in that we can go from pathogen to protein to vaccine candidate within a matter of weeks. The challenge with mRNA vaccines is that they are incredibly unstable. So we need to keep them at very cold temperatures in order to ship them around the country and deliver them to patients. DNA vaccines solve this problem because they are shelf stable. The challenge with DNA vaccines, though, is that they're a little bit too large to get into the cells. So what my lab is doing is developing a technique to help get these DNA vaccines into our cells. What we do is we deliver an electrical pulse, and these pulses help push the DNA vaccine candidates into the cells. So these vaccines are incredibly helpful for things like pandemic diseases like COVID-19, but also things like measles, influenza, and even cancer. The project that Ashley is working on this summer is to develop new protocols for helping to push these DNA vaccines into the cells.
So this summer, I've been working really closely with one of the PhD students in our lab, Robert Williamson. So his entire PhD is pretty focused on developing these DNA vaccines. This summer, we've been working on translating that from working in 3D cell cultures to actually an in vivo model. So we've been working with mice. As a veterinary student, it's been really cool to be able to come in and work on the animal handling and help Robert with the issues that we've been seeing when translating from a cell culture model into an animal model. So when you move into a whole animal model, it becomes a lot more complicated because of the variety of cell types you have, the complexity of the cell matrix, versus a very controlled single cell type in a collagen matrix within a dish, which is a lot easier to control. There's a lot less factors that you have to consider. Yeah, so I had a little bit of experience with research in undergrad, and I think something that drew me to the VSP program as a whole was the ability to get more involved with that. Something about research that's really compelling to me is the ability to make change on a broader scale. Obviously in clinical work, you're very hands-on, very tangible with a single patient, but the ability to develop new technologies that have the capacity to help millions of animals and humans alike is really powerful to me. It's been a lot of fun this summer. Like he mentioned earlier, our lab is about 14 deep right now. So it's been really great getting to know everybody. It's been really awesome because there's so many different projects going on. So even when I'm not actively working on experiments, I get to watch and learn from everyone with sort of different backgrounds and all of the different experiments we're doing in various animal models, as well as our equine patients. Those have been really exciting to watch.
So as my first year mentoring in the VSP program, it's been really exciting to have students who are prepared to do research come in and play a hands-on role in our experiments. So I think first day in the lab, Ashley was helping out with a procedure and just jumped straight in, and that was amazing. Typically when students come in, they're a little bit more timid, and it seems like with the veterinary students, they're really well prepared to just come in and start contributing right away.